Thank you so much. Good morning, everyone. Thank you all for joining the session this morning. I'd like to take a moment and thank the summit organizers for putting together such a fantastic program and allowing me this opportunity to present. So I know that many of you in the audience have an extensive background in genomics, but today I'd like to talk a little bit about how proteomics can be combined with genomics to provide a more holistic view of the patient. So I'll be talking a little bit about how proteogenomics can be a really useful tool for identifying subsets of patients who are already stratified with a given mutation, such as KRAS. So this provides a novel layer of information that can be really useful both for prognosis as well as treatment decision-making. So the talk that I'm going over today is entitled Real-World Data, and I'm going to talk about the importance of monitoring KRAS mutations, specifically in the blood. And just really briefly about myself, my name is Namratha, and I'm the Manager of Medical Affairs at Biodesix. Okay, so for those of you who may not be familiar with Biodesix, I just want to give you a little bit of an intro. So this is a data-driven diagnostic company, and the primary focus is really on lung cancer. So we have a streamlined system in place that's really aimed at both tumor profiling as well as identifying and understanding the host and immune profile using AI as well as machine learning. So we have 7 different commercially available diagnostic tests, and many of them have really quick turnaround times. Okay. So this is really the main assay that I'm going to be discussing today. This is a blood-based testing strategy, and it's called Biodesix Lung Reflex. And this is a blood test that has 2 different components to it. So you can see on the left-hand side here, there's a test called GeneStrat, and this is a genomic test. And the goal of this test is to identify actionable mutations in the blood. And this is using a technique called ddPCR or droplet digital PCR. And really, the goal here is to try to find therapies that targeted therapies based on actionable tumor mutations. So all of these are going to have an FDA-approved therapy associated with them. And I'd also like to note that this is a test that has a really quick turnaround time of 36 hours. And the reason I want to bring that up is because with many cancers, and certainly with non-small cell lung cancer, where most patients are diagnosed at later stages, primarily stage 4, it's really, really important to have a quick turnaround time for these patients because the median overall survival is quite low. So I'm going to be spending the first portion of this talk talking about the genomic test, GeneStrat, which is on the left-hand side. I do want to just quickly point out the VeriStrat test, which I'll come back to later, but this is the proteomic component of the of the Biodesix lung reflex system. Okay. So now I just want to take a moment and talk a little more about exactly where this test is being utilized. So here, of course, you can see a map of the US, and this is a test that's kind of being used all over the country. And I pointed out on the previous slide that this test has an average, a turnaround time of 36 hours. That's actually kind of the maximum time. And in a recent analysis that we did, we actually saw that the average is closer to 28 hours, so just over a day. Okay. All right, so I'm going to go over a quick introduction and then also tell you a little bit about KRAS mutations specifically with patients with non-small cell lung cancer. So when you have patients that have non-small cell lung cancer, or NSCLC, and they have KRAS mutations, generally these are patients that unfortunately have really poor prognosis. But of course, you can see on the right-hand side, this is a really exciting field, as I'm sure most people in the audience are aware. There are lots of novel KRAS targeting therapies that are coming out, and outcomes can certainly be improved by utilizing those. And of course, for the moment, I'm talking mostly about sotorasib and adagrasib. So, while these are really, really exciting, we still need a way to be able to figure out which patients need these drugs and which patients can be, which patients we can help by utilizing them. So having a way to really quickly identify the patients that are treatment eligible is really important. And our solution to that is to use a blood-based test. That, as I mentioned, it has a quick turnaround time, and the goal is really to expedite time to treatment and give these patients the best chance they have to try to fight their disease. Okay, all right. Okay, so now let's talk a little bit more in detail about KRAS mutations in non-small cell lung cancer. So you can see on the slide here, I've got a couple different pie charts, and what this is showing is that about a quarter of patients that have the adenocarcinoma histology within NSCLC harbor a KRAS mutation. And you can also see on the top right there, the most frequent mutation that we see for KRAS specifically is the G12C mutation. That tends to be the most prevalent. And unfortunately, having the G12C mutation tends to be associated with pretty poor prognosis, especially when it comes to chemotherapy. And you can also see on the bottom right, that's split up into never smokers and former and current smokers. And the prevalence of that G12C mutation really kind of comes from the smoking population. And in fact, with never smokers, the most common mutation that we see is G12D. But as I mentioned on the previous slide, the phase 2 data that we're seeing for some of the novel KRAS targeting therapies, such as sotorasib, are really exciting, with an objective response of 37% and an 80% disease control rate. So I'm really excited to see what's going to happen with these drugs going forward. So the GeneStrat test that I'm going to tell you about in the next couple slides identifies the 3 most common KRAS variants that we see within non-small cell lung cancer. So these are G12C, V, and D. Okay. So now I want to get into the test itself. So as I mentioned, the GeneStrat test, it's a genomic assay that utilizes ddPCR. And this is a slide that's showing some of the performance specifications for this test, some of the performance metrics. You can see on the left-hand side here, these are all of the mutations that are tested by, that this test actually looks at and identifies. And you can also see in the middle here, I've highlighted, oops, sorry about that. In the middle, I've highlighted KRAS, which has the 3 different variants that we measure. And if you look at the different columns on this slide, you can see that this is a test that has a really high clinical sensitivity. For KRAS specifically, it's 88%, but on the bottom row there shows the test that the combined variant results for all of the different mutations. And for that, you can see it's at 91%. And what's really exciting about this test is that it doesn't provide any false positives. So it has a perfect specificity. It's 100% across the board. And finally, the last thing that I want to point out is the concordance. So I'm sure many of you are probably aware in this field, it's really kind of considered that when you have tissue testing, that's kind of the gold standard. And it provides a lot of value to look at mutations specifically within the tissue. But given that this is a blood-based test, we wanted to see how well it does when in comparison to tissue. And we see that it has a really high concordance. So again, for KRAS, it's 96%. And for all of the different variants, it's 97%. Okay. So for the next couple slides, I'd like to talk a little bit more about the KRAS G12C variant in particular. So when we look at copy numbers for G12C, when we look at the mutation copies, what we see is that there's actually quite a different range, quite a large range, I should say, of copies that we see for these patients that ranges from, about 2 to 1,400. And this is also true when it comes to the minor variant frequency, which is shown on the right-hand side. And I specifically want to point out the percent MVF, minor variant frequency, because this is a way to kind of compare other techniques. So you can use this to try to compare how a ddPCR assay performs relative to another technique, such as next generation sequencing. And that's what I'm going to be showing in the subsequent slides. So our GeneStrat assay is being developed into an NGS test. And I'd love to share some of the data that we have related to NGS in the next couple slides here. Okay, so this this slide looks at concordance between these 2 assays. So what you're seeing here is the percent MVF for ddPCR shown on the x-axis on this slide, and for NGS on the y-axis. And what you can see here is the scatter plot and the best fit line associated with it. The R-squared value as well as the slope are really close to 1, so it's a pretty good fit. And you can see also that the sensitivity and specificity are really high. It's about 92% and 100% between the 2 assays. So it shows a really strong concordance between these 2 different tests. And also, I'd like to note that this particular graph that I'm showing you is specific to, it's not specific to anything. It's actually showing all of the different variants that we measure. But I wanted to also show a graph that looks specifically at KRAS G12C. And that's what's being shown on the right-hand side here. Again, I'm showing you the percent MVF for ddPCR and NGS. And again, you can see that both the slope as well as the R-squared value are really high. They're both pretty close to 1. And you can also see on the bottom right here that when you look at sensitivity, specificity, as well as concordance, for KRAS G12C specifically, it's 100% for all 3 of those. Okay, so as I'm sure you will all agree with me, genomic data is really useful when it comes to making treatment decisions. But as I mentioned, proteomics is also a really valuable tool. So we are currently conducting a registry study called the INSIGHT Observational Trial. And the goal of this is really to try to combine both of those elements to see how both the genomic as well as the proteomic components can be combined to provide a more, kind of holistic view and help to determine treatments in a real world setting. So that's really the goal of this study. And the next couple slides that I'm going to be showing you are related to our INSIGHT clinical trial. So I want to talk a bit about the study subjects and how the enrollment looks. Just a quick note, this is this particular slide is from our 3rd interim analysis that we conducted, where we had about 3,500 subjects that were enrolled. I would like to note that we have progressed since then, and right now we have about 43 subjects that have been enrolled, 4,300 subjects that have been enrolled. But when we had the 3,500 subjects, this is what the breakdown kind of looks like. Of those, just under 2,000 had more than a year of follow-up. So these are patients that had been in the trial long enough, and we'd collected enough data to be able to do some analyses here. And about a little more than 500 of those patients were early stage, and just under 1,500 were advanced stage NSCLC, so 1,464. And then from there, the breakdown goes is split between first line and higher line treatments. We had 877 patients with first line therapies. And of those, there's a split between the ones that received the ICI regimen, the immune checkpoint inhibitor regimens, that has 284 patients, and we had 392 that received just platinum-based chemotherapy. Okay, so now I want to talk more about the data from this trial and what we've seen thus far. And I should note these are pretty kind of hot off the press. This was actually presented at World Lung from the IASLC. This was actually presented about 2 weeks ago, so this is pretty new. And this is just showing the prevalence of KRAS mutations that we see across stages. So you can see on the y-axis there, on the x-axis there, it shows the disease stage going from 1A all the way to 4. And then we're measuring the percent KRAS mutation on the y-axis. So what you see is that certainly the prevalence of these different mutations, KRAS G12C, V, and D, it increases with stage. And you can also see that when you look across all different stages, the total percent of KRAS mutations that we see is just under 10%. Here it was 9.6%. And this is it's similar to what we see in the literature. It kind of mirrors what other liquid biopsy studies have also shown. Okay. Looking a little more closely at KRAS detection and the specific variants that are involved, again, you can see that just under 10% in both of these different cohorts. Just under 10% of the patients have KRAS detected. And here I'm showing 2 different cohorts. So on the right-hand side, this is the INSIGHT study that I just introduced. But on the left-hand side, this is actually a clinical use cohort. So this is more of like a real world setting with 25,000 patients. And here you can see, percent KRAS detected was 8.5% and 9.6% for the clinical use and INSIGHT cohorts, respectively. And then when we look more closely at the different types of variants, you can see that just about half have the G12C variant. It's just under half on the left-hand side and just over half on the right-hand side. And you can also see that when you look at the split between G12D and V, it's relatively 50-50 with the remaining patients. Okay. The last thing that I want to talk about with this particular set of data is related a little bit more with the covariates that we looked at. So we looked at three main covariates, stage, smoking status, and histology. And what you can see is that KRAS mutation prevalence tends to be higher in certain cohorts than others. So as I've kind of already alluded to, stage tends to be, later stage patients tend to have more of a prevalence in that, and specifically true when you look at stage 4 patients. It's also It's also It's also higher in prevalence in the ever smoking group relative to the never smokers. And finally, when it comes to histology, it tends to have a greater prevalence in the non-squamous histology relative to the squamous histology. We also did look at co-mutations with KRAS, but you generally don't see a ton of co-mutations in patients when you look at KRAS. And when we looked actually at our INSIGHT study, we didn't see any. So the far right of this graph is actually all from our clinical use cohort. And we did see a very small number of co-mutations, and these are with ALK, BRAF, and EGFR. Okay. All right, so now I want to come back to this slide that I showed you previously. So as I mentioned, I spent most of the talk so far discussing the left-hand side of this slide. And now I want to switch gears a little bit and talk about the right-hand side, which is the proteomic test. This is a test called VeriStrat. And this is a test that utilizes a proteomic signature that using machine learning and mass spectrometry. And basically what this test does is it identifies the presence of chronic inflammation in the blood. So when you have chronic inflammation in the blood, that generally creates this immunosuppressive environment, and that promotes really aggressive diseases in patients. So we see that patients that do have this aggressive disease, they tend to have worse overall survival. And this is a test that provides a binary result between VeriStrat good or VeriStrat poor. So patients that are characterized as VeriStrat poor do have that chronic inflammation, and that leads to them having a much more aggressive disease state relative to those who are VeriStrat good. The next couple slides, I'm going to be showing you a series of different Kaplan-Meier curves here, and all looking at overall survival. And I'm going to be showing you both how the genomic data look, essentially how you can split these patients based on KRAS mutations, and then introducing the proteomic classifier, VeriStrat, to see how we can further stratify these patients. So you can see on the left-hand, these are patients that have been stratified based on KRAS mutation status. And I want to note that these are patients that have received first-line platinum-based chemotherapy. And what you see is that when you look at median overall survival in these patients, patients that do have one of those three common KRAS variants tend to have much worse survival relative to those where KRAS is not detected. So you can see the hazard ratios and p-values listed on the bottom left of this slide. You can see there's quite a strong difference in stratification when you look at these two different patient subsets. But what we next wanted to do was look specifically at that cohort that did have a KRAS mutation present to see if we can further stratify those patients using the proteomic classifier VeriStrat. And that's what I'm going to be showing you on the right-hand side. So when you look specifically at the cohort that has the KRAS G12C, D, or B mutations, and then you use another classifier to further stratify them, you can see that the VeriStrat test does a pretty good job of being able to sort these patients out, and it identifies the ones that will have really poor survival, characterized as VeriStrat good, relative to the ones that have a much better survival, characterized as VeriStrat poor versus VeriStrat good. Okay, this next slide is looking at kind of a similar setup here, but this is looking at first-line ICI therapy. So this is immune checkpoint inhibitor therapy. And when we look at ICI regimens, KRAS is actually not a good stratifier in these instances. You can see these two lines pretty much overlap. There's really no difference between them. But again, when we look just at the patients that that had a KRAS mutation, and then we use VeriStrat to further stratify these patients, again, this test is able to further stratify, and it can pick out the patients that will have poor survival versus good survival. You can see here the median overall survival is comparing 6.3 months to a median survival that's not reached at 18 months. And finally, the last set of curves that I want to show you is looking a little bit more closely at immune checkpoint inhibition. So you can see on the left-hand side here, this is a cohort of patients that received ICI monotherapy. And on the right-hand side, I'm showing you a cohort of patients that received a combination. So this is ICI as well as chemo. And what you can see, again, when you basically introduce ICI, it seems like KRAS isn't able to effectively stratify these patients. These lines, once again, kind of lay on top of each other, and there's no significant difference between them. But what I'm going to show you next is, again, just that subset that has the KRAS mutation. And here you can see KRAS shows a very different story. So on the left-hand side, you can see there's a really strong difference between these two curves when you're looking at ICI monotherapy. There's quite a strong separation between these two with the hazard ratio of 0.22. But on the right-hand side, it looks a little bit different. So VeriStrat can still somewhat stratify these patients, but you can see that these lines are much, much closer together. So what this kind of indicates is that patients that have that VeriStrat poor status who also harbor a KRAS mutation seem to have better survival when given ICI as well as chemo relative to ICI monotherapy. Okay, so now I want to get to some conclusions and future directions. So as I hope I've illustrated in this presentation, blood-based testing can be really useful when trying to understand more about different biomarkers. And as I mentioned before, getting these molecular test results quickly is really, really important for these patients because, as you saw in the survival curves, there's not a lot of time that they have for median overall survival. So having expedited time to treatment is absolutely crucial for these patients. And of course, blood-based testing has the added advantage of being minimally invasive. So it's easy to implement for the most part. And it's also possible to use this technique to try to monitor treatment responses and also look for progression events or some sort of resistance development that might be coming along later in the timeline. And the real world data that I presented from our INSIGHT study, it shows really kind of the value of combining these two different aspects of a patient, both the genomics as well as the proteomics, to provide another layer of information that can be useful both for prognosis as well as for treatment decision making. And having having a glimpse at that patient's immune profile and looking at the proteomics is a really valuable addition. So the next thing that I want to talk about is kind of where we can take this into the future. And I want to show you a little bit about how you can utilize proteogenomics as a tool to really try to understand more about the biological underpinnings and get a better resolution of what we're seeing in terms of a patient's biology. So proteogenomics poses a very, very exciting opportunity to understand more about a patient's biology and to also improve the biological resolution, which I'm showing you on this slide. So the VeriStrat test that I spoke about today utilizes mass spectrometry, specifically multi-TOF mass spectrometry, which is shown at the top of this table. And this can be really useful when you're trying to understand details about different types of proteins that are involved. But kind of similar to traditional LC-MS, you do need to have some sort of like a deconvolution technique, such as depleting the abundant proteins or using antibody-based approaches to really be able to effectively utilize this. And you also have to have some understanding kind of a priori to get a really good understanding of the types of proteins that are involved. So there are other techniques that we can use and maybe combine to try to improve this resolution. So the next on this list is the protein set enrichment analysis, or PSEA. This is kind of similar to gene set enrichment analysis, which you may be a little more familiar with. And this provides another layer of resolution. So essentially, this looks at protein pathways, but it still relies on correlations, which may not tell you the whole story when you're trying to understand, really in depth of the biological underpinnings. So for that, we can use something called Shapley values. This is a really useful way to kind of break the black box. It's really hard to say together. So essentially, this is kind of like peeking into that black box of the machine learning algorithm and actually seeing what's going on inside and trying to explain that algorithm a little bit better. And essentially what it does is points out the relative importance of certain attributes of the mass spectra relative to other attributes. So kind of picks out the peaks that are the most important for a given patient. And from there, we can utilize more kind of advanced mass spectrometry techniques such as timsTOF. And this is another type of mass spec. And essentially, this is kind of the next generation of mass spectrometry. So unlike more traditional types of mass spec, this allows us to better understand the specific peptides that are used by these classifiers, especially if they're lower in abundance and might be harder to detect with the more traditional techniques. And finally, the best approaches often come while you combine the best parts of existing approaches. So at Biodesix, we're trying to do this really by utilizing the strengths of Shapley values as well as timsTOF, but also utilizing another technique called the Seer proteograph. And essentially, this is a way where you can use a nanoparticle that binds different proteins and creates something called a protein corona. And from there, you can use that to try to identify low abundance proteins. So essentially, by combining all of these different techniques, it yields a technology that's really sensitive and reproducible, while still examining the relevant peptides, regardless of their overall abundance. Okay, so with that, I'd like to send out a huge thank you to all of our Insight patients and our investigators, who have allowed us to conduct these analyses and, execute this clinical trial. And just a very, very big thank you to all of their work. And with that, thank you all so much for your attention, and I'm happy to take any questions.